A very warm welcome to the session on Golgi apparatus. Camillo Golgi in the year 1898 discovered an internal reticular apparatus in the nerve cells of the barn owl as a special cytoplasmic area impregnated with silver nitrate. These stained regions were termed as Golgi apparatus or Golgi body. Under electron microscope, it has been observed that these regions show flattened membranous vesicles. These are known as Golgi complex. Golgi complex is a differentiated portion of the endomembrane system which is morphologically very similar in plant and animal cells. It is specifically and temporally related to the endoplasmic reticulum on one side and to secretory vesicles leading to the plasma membrane on the other. Golgi complex plays as an intermediary in secretory processes. One quality of Golgi complex is lack of attached ribosomes. Most of the Golgi complex is surrounded by a zone from which most ribosomes, glycogen and mitochondria are absent, the so-called zone of exclusion. Some free polysomes have been observed at the periphery of the Golgi complex. Moving ahead, we will now study electron microscopic structure of Golgi complex. Dalton and Felix in 1954 were first to reveal the electron microscopic structure of the Golgi complex. In general, three membranous components are observed under the electron microscope. They are flattened sacs or cisternae, clusters of tubules and vesicles of about 60 nanometer, and large vacuoles filled with an amorphous or granular content. Cisternae. The Golgi cisternae are arranged in parallel way to form stacks. Cisternae are separated by a space of 20 to 30 nanometer which may contain rod-like elements or fibers. Often, the cisternae are arranged concentrically with a convex or concave face. There may be from 3 to 7 of cisternae in most animal and plant cells. In certain algae, there may be as many as 10 or 20 cisternae. Each stack of cisternae forming a dictyosome is a polarized structure having a proximal or forming face, generally convex and closer to the nuclear membrane or the endoplasmic reticulum, abbreviated as ER, and a distal or maturing face of concave shape which encloses a region of large secretory vesicles. This polarization is often referred to as the cis-trans axis of the Golgi complex. The forming phase, that is cis phase, is characterized by the presence of small transition vesicles or tubules that converge upon the Golgi cisternae, forming a kind of fenestrated plate. These transition vesicles form blebs from the ER and migrate to the Golgi, whereby coalescence they form new cisternae. New cisternae are formed at the proximal end by mechanism of membrane flow and thus compensate for the loss of released secretory vesicles at the maturing phase. Associated with the maturing phase, there is secular structure rich in acid phosphatase called as capital GERL, that is abbreviation for Golgi complex, endoplasmic reticulum and lysosome system. GERL has been interpreted as region of smooth ER near the Golgi involved in the production of lysosomes. Tubules and vesicles. From the peripheral area of cisternae arise a complex anestomizing flat network of tubules of 30 to 50 nanometer diameter. The vesicles are small droplet-like sacs which remain attached to tubules at the periphery of the cisternae and may show continuity with the cisternae. The small vesicles arise from the cisternae by budding or pinching off. These are 40 to 80 nanometer diameter vesicles, which are of two types. They are smooth vesicles and coated vesicles. Smooth vesicles or secretory vesicles. The smooth vesicles are of 20 to 80 micrometer in diameter. They contain secretory material and are budded off from the ends of the cisternal tubules within the net. Coated vesicles. The coated vesicles are spherical protuberances about 50 micrometer in diameter, having a rough surface. They are found at the periphery of the organelle, usually at the ends of single tubules. Large vacuoles. The Golgi vacuoles are large, spacious, rounded, sac-like structures occurring at the distal ends of cisternae. They are produced by vesiculation of sacules of cisternae. Furthermore, we will discuss functions of the Golgi complex. Golgi complex plays an important role in the storage, packaging and secretion of certain cell products. It is involved in the formation of lysosomes and other enzyme containing cellular inclusions. It is also involved in the formation of secretory granules in cells such as those found in the pancreas, the pituitary and mammary glands, and in the mucus secreting glands of the intestine and in many other cell types. The Golgi complex is mainly concerned with formation and packaging of material for export from the cell through the plasma membrane by reverse pinocytosis. In this way, cell wall material is deposited in plants and secretory proteins, digestive enzymes and the matrix of the connective tissue are transported in animal tissue. The Golgi complex represents a special membranous compartment interposed between ER and the extracellular space. Through this compartment, there is continuous traffic of substances which have been synthesized elsewhere but are modified and transformed while transported. This process also involves the flow and differentiation of membranes. Within the membranes of the Golgi complex, a vast diversity of materials may be found, varying from simple fluids to macromolecules. Every type of macromolecule is transported through the Golgi and secreted, and there is a continuous and fast turnover of the Golgi membranes. Some of the most important functions of Golgi complex are as described here.
Again.
Synthesis of glycosphingolipids and glycoproteins, major function of the Golgi complex. The Golgi complex plays a major role in the glycosidation of lipids and proteins to form glycosphingolipids and glycoproteins. The carbohydrate prostatic groups of both these complex substances are added in a sequential manner in the Golgi complex by the action of the various glycosyl transferases. The enzymes are responsible for transferring a sugar residue either to an amino acid residue of a protein or to another carbohydrate in a glycoprotein or glycolipid. From studies using radioactive precursors, example galactose, fucose and sialic acid, the following biosynthetic scheme has emerged for the types of secreted glycoproteins. The protein backbone of the glycoprotein is synthesized by ribosomes and then the monosaccharides are added one by one as the protein moves through the channels of the ER and Golgi complex. In this process, the glycosyl transferases that are bound to the Golgi membranes play an important role. The enzymes adding the oligosaccharide core containing N-acetyl glucosamine and mannose are in part assembled in the rough endoplasmic reticulum. The fucosyl and the diallyl galactosyl N-acetyl glucosamine termini of the prostatic group are incorporated in the Golgi complex by the corresponding transferases. There are three main patterns of incorporation of various precursors into glycoproteins. When D-mannose is used as precursor, the rough ER, that is RER, is the first to be marked and then the other components. When sialic acid, L-fucose or D-galactose is used, the Golgi complex is the first to be labeled. With D-glucosamine, both the RER and Golgi complex are simultaneously labeled. Radioautography revealed that H3-fucose does not enter into mucopolysaccharides and fucose is present in glycoproteins only in the terminal group. In intestinal cells, 2 minutes after the injection of the H3-fucose, the labeling is almost exclusively in the Golgi complex. Then there is migration of the glycoproteins to the sides of the cell and into the apical cell membranes, where it is concentrated 4 hours after injection. The intracellular migration of the glycoproteins is carried out by small vesicles formed in the Golgi complex. In liver cells, H3-fucose is first incorporated into the Golgi and later on is found in lysosomes, in the blood plasma and on the cell membranes. The polypeptide backbone of the glycoprotein is synthesized on the ribosomes and side chains of mannose and N-acetyl glucosamine are added in ER, whereas the terminal side chains of galactose, fucose and sialic acid are added in the Golgi complex. The substrates for glycosyl and sulfate transferases are not the sugar moieties or the sulfate. The true donor molecules are sugar nucleotides rich in energy such as guanosine 5' diphosphate fucose or mannose, uridine 5' diphosphate glucose, galactose or N-acetyl glucosamine. The adenosine 3' phosphate 5' phosphosulfate is the donor of the sulfate moiety. All these compounds are synthesized in the cytosol and traverse the membrane to become substrates of the enzymes which have their active centers at the luminal side of the Golgi vesicle. Secretion, the main function of the Golgi complex. ER and the Golgi complex are more directly involved in the synthesis, transport and release of macromolecules from the cell. Cell secretion is not confined to animal cells. Plant cells also secrete polysaccharides and proteins to build their cell walls. The enzymes of lysosomes and peroxisomes of animal and plant cells are produced by a kind of secretory process. Cell secretory activity appears even in the prokaryotic cells, producing the cell wall and secrete a variety of enzymes to the medium. Secretion may be continuous, the secretion products being discharged without storage, that is glycoproteins by the liver cells, antibodies by the plasma cells. In these cells, the secretion produced is not accumulated into special storage granules and the release of secretory materials is more or less simultaneous with the synthesis and intracellular transport of these substances. Secretion may be discontinuous, with storage in secretory or zymogen granules, for example in the pancreas, parotid glands and others. In these cells, secretory cycle is specially timed so that the synthesis and intracellular transport are followed by the accumulation of the secretion products in special storage granules, which are finally released to the extracellular space. These products may be dense and refractile granules, vacuoles, droplets or other substances having a definite location in the cell and have characteristic histochemical reactions. The dense secretory granules containing enzymes, generally in an inactive form, that is proenzyme, are called the zymogen granules. Endocytosis and recycling of membranes of secretory granules. A pancreatic cell in which protein synthesis has been inhibited can go through an entire secretory cycle of transport, concentration, storage and release. This suggests that within the period of time of one cycle, that is 60 to 90 minutes, the synthesis of new membrane proteins is not required. The intracellular membranes have a much longer half-life and are reutilized extensively during the secretory process. The membrane of the zymogen granules may be regarded as a vacuole that shuttles between the Golgi complex and the cell surface. In the removal of excess membrane from the apical region of the cell, it has been suggested that patches of membranes are invaginated from the surface as small vesicles that move back into the Golgi region to be reutilized in the packing of more secretion. Thus, the process of exocytosis is coupled with that of endocytosis. The specificity of this recycling process depends on the formation of coated pits and vesicles which separate the membrane patches from the plasma membrane by mechanism of selective endocytosis. The morphological and histochemical evidences indicate a possible heterogeneity of function within the Golgi complex. For example, galactose residues labeled by a specific lectin complexed to gold particles are localized in the trans region of the Golgi complex, while the cis region is unlabeled. In the same cells, thymine pyrophosphatase is localized in similar stacks of Golgi membranes. The co-distribution of these two enzymes suggests the existence of functional compartmentalization in the Golgi complex and favors a model of glycosidation in which two enzymes act in concert. Using monoclonal antibodies and electron microscopy, it has been found that N-acetyl glucosamine transferase 1 occurs in medial cisternae of the Golgi. Additional evidence indicates that the compartmentalization of the Golgi complex is provided by the presence of a gradient of cholesterol from the cis to the trans stacks. The Golgi complex have at least 3 distinct sets of cisternae besides the GERL region. If each cisternae has a specific function, then the traffic between the cis and trans side would be affected by vesicular carriers that operated from the dilated rims of the cisternae. The Golgi membrane proteins and specific enzymes are in fixed positions within the Golgi complex, but those proteins that enter the cisternae may diffuse throughout the entire complex by this carrier mechanism. The proteins transported by small vesicles from the ER are received at the cisternae and they exit from the trans cisternae at the opposite end of the stack. Lipid packaging and secretion. The electron microscopic and autoradiographic studies of stain and stain revealed the role of the Golgi complex in lipid secretion. Fatty acids and monoglycerides are used in the synthesis of lipids. The epithelial cells of intestine secrete chylomicrons which contain lipids in the form of lipoproteins. Labeled palmitate and glycerol were injected into fasted and ethanol treated rats. First, the labeled material, that is triglycerides, was seen in ER, then in storage lipid droplets, and finally in the Golgi complex about 10 minutes later. The ER is involved in triglyceride synthesis. The vesicles derived from the Golgi complex is involved in adding the carbohydrate components of chylomicrons. The overall role of the Golgi complex in lipid metabolism is the concentration and modification of secretory material. These changes convert the lipid droplets into chylomicrons. The Golgi complex also provides a membrane for covering of lipid to be released from the cell. Insulin biosynthesis. The role of Golgi complex in insulin biosynthesis has provided an excellent example of the molecular processing of secretion. The many secreted proteins are first synthesized as biologically inactive precursors which are activated later. Activation consists of removal of a portion of the polypeptide chain and occur at different sites. The zymogens secreted by the pancreas are activated extracellularly, that is, after the release of the secretion. Various polypeptide hormones are produced as inactive prohormones which are then activated intracellularly by the converting enzymes that are proteolytic enzymes present in the Golgi complex. Examples of proproteins are proalbumin, proparathyroid hormone, proglucagon, progestin and proinsulin. The insulin produced by the beta cells of the pancreatic island has a molecular weight of 12,000 and two chains, the A chain of 21 amino acids and the B chain of 30 amino acids. The chains are linked by two disulfide bonds. The insulin mRNA with its B and A regions, that are cistrons, code for the B and A chains. In addition, the mRNA contains a pre-region which codes for the signal peptide, a C segment, that is a connecting segment, and a poly A tail. This mRNA contains information coding for a polypeptide chain of 330 amino acids. The complete translation of this mRNA on free ribosomes gives rise to pre-proinsulin. In the ER, the pre-region, that is a signal peptide of 23 amino acids long, is removed by the signal peptidase. The completed proinsulin chain is then activated in the Golgi through the removal of the C peptide, that is connecting peptide, by the converting enzyme and is packaged as active hormone within the secretion granule. The GERL region and lysosome formation. Golgi membranes are involved in the formation of the primary lysosomes. This process has the sequence, that is synthesis, aggregation, transport and packaging of the enzymes. The ribosomes synthesize lysosomal hydrolases. These hydrolytic enzymes then enter the ER. The ER forms small vesicles of these hydrolases by blebbing. The vesicles are transferred to the Golgi complex. The cisternae of the Golgi complex in turn form vesicles. The primary lysosomes fuse with pinocytic vesicles or with autophagic vesicles and form secondary lysosomes. D. Dewey suggested that the Golgi vesicles transfer acid hydrolases to food vacuoles, converting them into digestive vacuoles. Lysosomes can also be formed from the ER without the Golgi complex, as in liver cells and in neurons. Because of the close functional relationships between the Golgi complex, ER and lysosomes, Novikov has denoted it as the GERL system. In the maturing phase of the Golgi is the GERL region, in which acid phosphatase, a characteristic lysosomal enzyme, is present. This region has been implicated in the formation of primary lysosomes. There is some kind of traffic control which exists in the Golgi complex to direct the various products to their final destination, that is, to the zymogen granules or the lysosomes. Novikov suggested that the GERL of the Golgi complex is also involved in the formation of melanin granules, in the processing and packaging of secretory material in endocrine and exocrine cells and in lipid metabolism. GERL, also known as the transreticular Golgi, is the site of the cell involved in the sorting out of the proteins. Acrosome formation. There is a close association between the Golgi complex and acrosome formation. The acrosome of a spermatozoon is derived from the Golgi complex of a spermatid. The Golgi complex of an early spermatid consists of a series of membranes arranged concentrically around an aggregation of small vacuoles. During acrosome formation, one or more vacuoles start enlarging, and inside the vacuole appears a small dense body, the proacrosomal granule. The contents of vacuole and granule give a positive staining reaction for mucopolysaccharides. The vacuole enlarges in its volume by fusing with other small vacuoles containing proacrosomal granule and become closely applied to the forwarding tip of the elongated nucleus. The granule increases further and becomes the acrosomal granule, which forms the core of the acrosome. The vacuole loses its liquid contents and its wall spreads out over the acrosomal granule. Front half of the nucleus, covered with a double membranous sheath, that is plasma membrane and membrane of acrosomal vesicle, forms the cap of spermatozoon. The remaining parts of Golgi complex are gradually reduced and ultimately discarded from sperm as Golgi rest together with some cytoplasm. Another function of Golgi complex is plasma membrane formation. It has been shown that secretory granules originating from the Golgi complex fuse with the plasma membrane during exocytosis. The membrane of the granules become incorporated into the plasma membrane and thus contributes to the renewal of the membrane constituents. The Golgi complex is involved in the synthesis of the carbohydrate components of the plasma membrane. The tritiated sugars, first localized at the Golgi complex, migrates to the cell surface and in the cell coat. Secretory glycoproteins are released into the extracellular space after fusion of the secretory vesicle membrane with the plasma membrane. The Golgi complex plays an important part in the biogenesis of the plasma membrane by supplying it with glycosidated molecules. Plant cell wall formation. Plant cell walls are made up of fibrils which predominantly contain polysaccharides, some lipids and proteins. The polysaccharides are formed in the Golgi complex and transferred to the new cell wall which is being formed. The Golgi complex also contributes the substances, pectins and hemicelluloses which form the matrix of the cell plate. Another function of Golgi complex is neurosecretion. A study of neurosecretory cells of invertebrates and vertebrates has revealed that neurosecretory granules lie close to the Golgi complex. The neurosecretory material is synthesized in the ribosome and is then transferred to the Golgi complex, where it undergoes packaging to form neurosecretory granules. Pigment formation. The GERL of the Golgi complex is also involved in the formation of melanin granules. In many mammalian and cancer cells, the Golgi complex is involved in origin of pigment granules, that is melanin. The Golgi complex has also been associated with pigment formation in retinal pigment epithelium of chick embryos, in the test cells of the ovaries of certain tunicates, and in the oocytes of the salamander. The last function of Golgi complex is formation of yolk substances and cortical granules of oocytes. During oogenesis, the Golgi complex is involved in the formation of yolk. In the young ovum, it breaks down into pieces and arrange themselves to form bulbianic vitelline rings or yolk nucleus. Soon, this arrangement is lost and the yolk particles appear in the cytoplasm. Lastly, we summarize the session. Golgi apparatus appear to resemble a small stack of flattened sacs. It assembles the protein products made by ribosomes into the usable proteins, and these finalized proteins are then packed into special sacs and are sent out to their final destination. Thus, Golgi bodies are frequently described as assembly lines or shipping and receiving facilities. Thank you.